Okay, so the sexual reproduction of in flowering plants, right? So you see so in here uh, so pre-fertilization, that is structure and events. Okay, the stamen, microsporangium and pollen grain. Stamen has filament and anther. Okay, the proximal end of filament is attached to the thalamus or the petal of the flower. Okay, the number and length of stamens are variable in number of different species, that is salvinia. Okay, and then a typical angiosperm anther is bilobed with each lobe having two theca, that is dithecous. Okay, then the anther is four-sided, that is tetragonal structure. It has four microsporangia located at the corners, two in each lobe. Okay, the microsporangia develop further and become pollen sac. So anther has the like four microsporangia that forms the microsporangia develops into pollen sacs. Okay, and then inside the pollen sacs there is like formation of microspore through the sporogenous tissue. It is diploid, so microspore mother cell, it is diploid, it goes to meiosis and it forms like microspore. This microspore after maturation it becomes pollen grain. Okay, so this is the structure of microsporangium. As you know, it has four walls: epidermis, endothecium, middle layer and tapetum. Tapetum is for nourishment of pollen grain. Epidermis, endothecium and middle layer is for like protection and help in dehiscence when the anther is mature. Okay, so it has like sporogenous tissue at the center. Macrosporogenesis, they undergo meiosis and they form microspore tetrad, and ploidy is haploid. So there is like pollen or microspore mother cell. Okay, now PMC, pollen mother cell, it is diploid, it undergoes meiosis through microsporogenesis. Okay, and it forms cluster of four cells, that is microspore tetrad. And as the anther matures and dehydrate, the microspore dissociate from each other and develop into pollen grains, right? Now pollen grains, so pollen grain represents the male gametophyte. Just like in ovule there is embryo sac, embryo sac is the female gametophyte. So pollen grain is 25 to 50 micrometers in diameter. It has exine and intine. Intine made up of cellulose and pectin. Exine made up of sporopollenin. It is very hard. It is not degraded. Also, intine is continuous. Exine is discontinuous. It has germ pore in which pollen tube grows. So through which the pollen tube grows, and a pollen grain has at maturity it has two cells, that is generative, that is vegetative, and that is generative. So generative cell, it is bigger, has abundant food reserve material and large irregular shaped nucleus, and generative cell is small, floats in cytoplasm of the vegetative cell. And this generative cell, it is spindle shaped with dense cytoplasm and a nucleus. So 60% of angiosperm, it is the pollen grain shed at two cell stage. In the remaining, like the cell divides mitotically to give rise to two male gametes before pollen grains are shed, that is three cell stage. Now pollen grains causes severe allergies and bronchial afflictions, okay, leading to chronic respiratory disorders, so asthma and bronchitis. Now parthenium and carrot grass that came into India is contaminated with imported wheat, okay, has become the problems. It causes the pollen allergy. Now pollen allergy, these are rich in nutrients, so it uses as tablets and syrups. Now athlete to increase their performance and race horses. Okay, now pollen viability, like within and 30 minutes some cereals, rice and wheat, and for months like members of Rosaceae, Leguminosae and Solanaceae. Okay, and minus 196 degrees Celsius you can store the pollen grains in liquid nitrogen. And pollen also used as pollen banks in crop breeding programs. So pistil, megasporangium, ovule and embryosac. So pistil, that is carpel, it has stigma style and ovary. Now monocarpellary, multicarpellary, syncarpous. Syncarpous, that is fused. Multicarpellary, they are can be free. Monocarpellary, single. Okay, now apocarpous. So ovary is the basal part, stigma, ovary is the basal part, right? There is an ovarian cavity inside the ovule. Now the placenta, it is cushion like structure where the ovule is attaches, and ovule has the stalk, that is funicle. and funicle and ovule has a scar. No, that is hilum is the junction between funicle and ovule. Okay. Then the multicarpary, that is syncarpus, example is papaver, and multicarpary apocarpus gynoecium, that is michelia. So apocarpus michelia and syncarpus papaver. Now we study the anatropous ovule. So this is anatropous ovule. So you can say like funicle, then hilum and microbial. So junction between ovule and funicle is hilum. Then there is outer integument, inner integument. There is nucellus, and then is embryo sac. Opposite is the chalazal pole. As you can see, sigma style and syncarpus over there. Now arising from the placenta are the megasporangia. These are the ovules. Okay, and the number of ovules in maybe one, that is wheat, paddy, mango, or papaya, that is wheat, paddy, and mango, and papaya, watermelon, orchids. They are the megasporangia, that is ovule. Now ovule is a small structure attached to the placenta by means of stalk, that is funicle. So one or two protective envelopes called integuments. As I show you, like so the integuments, these are protective in nature. They encircle except at the tip where a small opening, that is micropyle, is organized. Opposite to micropyle and is a chalazal that represents the basal part of the ovule. So enclosed within the integuments is a mass of cells called the nucellus. Now cells of the nucellus are abundant rhizophore materials. Located in the nucellus is the embryo sac or the female gametophyte, and all generally has a single embryo sac from megaspore. So megasporogenesis, the process of formation of megaspore from the megaspore mother cell is called megasporogenesis. Now over generally differentiate a single megaspore mother cell, that is MMC, in the micropyle region of the nucellus. Now it is large cell, it undergoes meiotic division, it forms like the megaspore. Now female gametophyte, in majority of flowering plants one of the megaspores is functional while the other is rigid. Only the functional megaspore develops into female gametophyte, that is embryo sac. So this method of embryo sac formation from a single megaspore is monospory development. So what will be the priority of the nucellus? Nucellus, I think it is haploid or diploid, I don't know. Go and search on the Google. The megaspore mother cell, it is diploid. The functional megaspore, it is haploid. Female gametophyte is a haploid. Okay, so this is it. Like you can see in here, like synergids, egg, central cell, two polar nuclei, antipodal cells. Then micropyle in synergids has the filiform apparatus, then egg, then central cell, then it has the polar nuclei, then antipodal cells, then chalazal end. You see there is a formation of megaspore dyad, then megaspore tetrad, 3 gets degenerate, 1 is functional. Okay, now 2 nucleate, then 4 nucleate, then 8 nucleate, undergoes 3 mitotic divisions. So after the 8 nucleate cells were, so it is strictly the nuclear divisions are not followed immediately by cell wall formation. Okay, so so after the 8 nucleate cell walls are laid down, leading to the organization of the typical female gametophyte or embryo sac. Okay, 6 of the 8 nuclei are surrounded by cell walls and organized into cells. The remaining 2 nuclei called polar nuclei are situated below the egg apparatus in the large central cell. Okay, now 3 cells group into 3 cells, that is 2 synergids and 1 egg cell. They make the egg apparatus. Now synergids have special cellular thickenings at the micropyle tip called the filiform apparatus, which play an important role in guiding the pollen tube into the syncytium. Now 3 cells at the chalazal end called the antipodal cell. Last central cell, as mentioned, has 2 polar nuclei. Thus a typical angiosperm embryo sac at maturity, though 8 nucleate and 7 cell. Now pollination is a mechanism to now transfer of pollen initiated from the anther to stigma of a pistil is termed as pollination. Now kinds of pollination. So depending on the source of pollination can be 3 types: the autogamy, deuteronogamy, and xenogamy. So same flower, same plant is the autogamy, and flowers are bisexual in celastogamous flowers. Okay, and autogamy has like celastogamous and chasmogamous flowers. They are open all the time. Celastogamous, they are always closed. Examples are viola, that is common, passive oxalis, that is camelina. So they produce 2 types of flowers, right? So they produce 2 types of flowers. Okay, in which like I mean stigma and pollen, and stigma and pollen, they lie close to each other. So this is like, and celastogamous flowers are invariably autogamous, as there is no chance of cross pollination landing on the stigma. So celastogamous flowers produce assured seed set even in the absence of pollinators. So it is disadvantage, it is advantage also. So deuterogamy, that means different flowers of the same plant. Okay, deuterogamy is cross pollination, genetically it is similar. Okay, now xenogamy is like different flowers of different plants, I mean dioecious condition, right? So pollination brings genetically different types of pollen grains to the stigma. Now agents of pollinations like abiotic and biotic. So in abiotic it is wind and water, and one biotic, that is animals. Okay. Now the majority of plants use biotic agents, and mostly they are the animals in which the bees are an insect, and in which bees are the dominating pollinating agents. Okay. Now in wind it is a chance factor, and pollens produced in numerous amount, and compared to the oils, and oils generally one in number. Okay. So this is it. And in pollination by wind is more common amongst the abiotic pollinators. Wind pollination also requires that the pollen grains are light and non-sticky. The pollen grains are light and light, so they can transport into. So now well exposed stamens and often feathery stigma to trap the airborne pollen grains. Okay. Now wind pollinated flowers have single ovule in each ovary, and numerous flowers packed into an inflorescence. Okay. Now the corn cob, you can see the like stigma styles. These are like stigma style. These are the stacels, and they used to trap the pollen grains. Okay. The wind pollination is quite common in grasses. So wind pollination is quite common in grasses, right? Now pollination by water is quite rare in flowering plants. Is limited up to 30 genera, mostly the monocot. Okay. Because the water is always restricted. Okay. For algae, bryophytes and pteridophytes, this is why they are restricted in places. Okay. Because water for transport of fertilization, all that. So examples like Vallisneria hydrilla, these are fresh water. Several marine seagrasses such as Zoosterum. Now not all aquatic plants use water for pollination. Okay. Now majority of aquatic plants such as water hyacinth and water lily emerge above the level of the water and pollinated by insects or wind, as in most of the land plants. Now Vallisneria, the female flower reached the surface by long stalk, and the male flowers or pollen grains are released onto the surface, and they are. So this is how like. And and now the Zoostera, that is sea grasses. The